Hello students, welcome to my channel Dr. Deepa Dahake's NEET Biology, and I am Dr. Deepa Dahake, Director at BIOZONE CLASSES, Jabalpur. In today's session, we are going to study about the most important topic and most important unit we begins here, that is from 11th syllabus. Plant physiology is a 4th unit, and in plant physiology today I am going to explain you respiration in plant, and first we deals with the most important topic of that chapter, that is glycolysis. But before beginning with session, मैं अब आपको लास्ट सेशन में जो क्विज सेक्शन हुआ था उसके आंसर्स प्रोवाइड करूंगी. देन आई विल आस्क द 3 more questions and then we begins with our session. So the answers for the last session, फर्स्ट क्वेश्चन का आंसर है आपका आंसर बी, दैट इज 50% रेसेसिव. यहाँ पे देखिए स्टूडेंट क्वेश्चन क्या था. क्वेश्चन इज व्हेन स्मॉल टी इज मेट्स विद कैपिटल टी एंड स्मॉल टी, and 4 options are provided over there. So small t is produces a similar type of gametes, but when capital T and small t we discuss, so it produces 2 different types of gamete because this is a hybrid. So whatever progenies are produces, that you remember, the 50% progenies are dominant and 50% progenies are recessive. In option, only correct option, one correct option is there. The 50% progenies are recessive. For dominant, D option provide था all dominant. So all dominant कब होंगे आपके? When this parent is capital T, then only the all progenies are dominant. Whenever the heterozygous parent crossed with homozygous recessive, ये कंडीशन याद रखिएगा, 50% डोमिनेंट एंड 50% रिसेसिव. सो फॉर दैट यू रिमेंबर करेक्ट आंसर आपका क्या है? करेक्ट आंसर इज बी. फॉर क्वेश्चन सेकेंड करेक्ट आंसर इज सी, एंड फॉर द क्वेश्चन 3 correct answer is D. So now these are the 3 answers for last session quiz, and now I am coming towards today's quiz section, and I kindly request you all provide your answers in comment box, so the queries and जो आपके doubts हैं, those doubts will be clear. Okay, so the questions for today's quiz are: question 1, which of the following most appropriately describe hemophilia? A. Recessive gene disorder. B. X-linked recessive gene disorder. C. Chromosomal disorder. D. Dominant gene disorder. Question second, number of bar body present in each somatic cell of female is A1, B2, C3, and D option is 4. Question third, Down syndrome in humans is due to option A, 2 Y chromosomes, B, 3 X chromosomes, C, 3 copies of chromosome 21, and D option is monosomy. Students, let's begin with glycolysis, one of the most important topic of the plant physiology unit from 11th syllabus. But before begins with session, I kindly request to all new students that subscribe my channel, like my videos, and comment for your queries and doubts, as well as provide the answers for the today's session. Okay, so let's begin with today's session. Students, let's begin with glycolysis. First यहाँ पर हम देखेंगे कि why we are study about the glycolysis, because this is one of the important phenomena and it is universal pathway for the breakdown of glucose. And during this pathway when we study, so one molecule of glucose is break down into 2 molecules of pyruvic acid. Glucose is a 6 carbon sugar, and this 6 carbon sugar, you remember, when undergoes in glycolysis, so it will break down into 3 carbon compound, that is the 2 molecules of pyruvic acids are formed. For your upcoming NEET examination as well as for the board purpose also, this glycolysis is important. So again you remember here, the glycolysis is important phenomena, and number of questions are related with the enzymes. So as you observe this chart, So two predominant enzyme which multiple time asked in competitive exam. So you remember the functions of these enzymes as well as how many reactions are take place in glycolysis, what are the steps in glycolysis which are responsible for energy generation, और ऐसे कौन से स्टेप है glycolysis में where the ATP is required for the phosphorylation. So that part you remember, यहाँ पर आपको study करना है. So that's why we now coming towards the study of glycolysis. अब हम glycolysis start करते हैं. So glycolysis you remember it is a Greek term and it is made by glycase as well as lysis. Glycase का meaning होता है breakdown of, glycase means sugar and lysis term stand for the breakdown. That means the process or the pathway responsible for the breakdown of sugar. What kind of sugar? So you simply remember a monosaccharide glucose. So when a glucose is undergoes in pathway and break down into two molecules of pyruvic acid, that pathway is called as glycolysis. The glycolysis you remember, glycolysis is also called as EMP pathway. You remember this another term for or another name, synonymous name for the glycolysis, EMP pathway, because the scientists Imbedin, Meyerhoff and Parnas, these three scientists first time provide this pathway for the anaerobic respiration. That's why this pathway is also called as EMP pathway. Now when we study about the organisms, so two types of organisms basically we study, or two types of individual, aerobic and anaerobic. So in anaerobic individual, you remember this is the one and only pathway for cellular respiration, that is the glycolysis. Whereas in aerobic में अगर आप देखते हैं, so in aerobic organisms or in aerobic cells, first glycolysis is take place and then after the aerobic pathway, that is Krebs cycle आप study करते हैं. So glycolysis in aerobic individuals followed by the Krebs cycle. But in anaerobic cells or anaerobic individual, this is the one and only pathway you remember for the breakdown of glucose or the metabolism of the glucose. Secondly, you remember it is completed. What is the site of glycolysis? When we study about the site of glycolysis, so you remember the phenomena or the pathway of glycolysis is completed in cytosol. And secondly, what is the net gain of glycolysis? Net gain means when the glycolysis is completed and glucose is break down into pyruvic acid, then how many number of ATP are synthesized? So net gain you remember, total gain of glycolysis is 8 ATP. So that we study when we study about the entire pathway of the glycolysis. But for objective purpose and NEET exam you remember the net gain of glycolysis is 8 ATP. And again ये already discuss कर लिया है हम लोगों ने कि anaerobic organisms में this is one and only path for the glucose metabolism, whereas in aerobic organisms or aerobic cells, glycolysis as well as Krebs cycles are responsible for the complete metabolism of glucose. Now I come towards the pathway. So it is a simply you remember when we study about the glycolysis, so you remember 10 enzyme catalyzed reactions are take place, and with the help of this series of 10 enzyme catalyzed reaction, the one molecule of glucose you remember a 6 carbon sugar or hexose sugar, and that 6 carbon sugar with the help of 10 enzyme catalyzed reaction get broken down into two molecules of pyruvic acid, that is a 3 carbon molecule. Now how does this pathways take place? Let's begin. Here you see glucose first undergoes in conversion of glucose 6-phosphate with the help of glucokinase or hexokinase enzyme. This is the process or this is the reaction where the first molecule of ATP is broken down and it break into ADP and inorganic phosphate. This inorganic phosphate is used for the formation of glucose 6-phosphate. In second reaction of glycolysis, the second reaction of glycolysis is isomeric reaction. The glucose 6-phosphate gets simply isomerized into fructose 6-phosphate. So here you remember the glucose 6-phosphate convert into fructose 6-phosphate. For NEET exam you remember glucose is a aldose sugar and fructose is a ketose sugar. So both C6H12O6 ही लिखते हैं, but structural formula if you go, so glucose आपका है aldose sugar and fructose you remember it is a ketose sugar. Now in third reaction of the glycolysis, the fructose 6-phosphate convert or change into fructose 1,6-biphosphate with the help of enzyme fructokinase, or simply in some of the book, reference book, you find as a hexokinase. Okay. Now again the second molecule of ATP is break into ADP and inorganic phosphate. Both the reactions where the ATP is break into ADP and inorganic phosphate, for that reaction magnesium ion is essentially required. So that part also you remember. And then this fructose 1,6-biphosphate now further undergoes in the next reaction. This is the most important reaction, and this enzyme, you remember the activity of aldolase enzyme, because this is the reaction of glycolysis in which the 6-carbon fructose 1,6-biphosphate break down into 3-carbon compound. 2 molecules of 3,3-carbon compounds are formed. That the 1-carbon compound is glyceraldehyde 3-phosphate, and the second 3-carbon sugar is formed or compound is formed, that is called as dihydroxyacetone phosphate. So you remember the enzyme aldolase activate on fructose 1,6-biphosphate. So what is the substrate for aldolase? The substrate for aldolase is a fructose 1,6-biphosphate. When aldolase is activating on fructose 1,6-biphosphate, so it will produce as a 3-carbon compound, glyceraldehyde 3-phosphate. Again remember, it is a triose sugar. So this triose sugar or 3-carbon sugar is a aldose sugar, and DHAP or dihydroxyacetone phosphate is a ketose sugar, but both the sugars are of 3-carbon. That's why they are called as triose sugar, and triose sugar, you remember, they are placed in monosaccharides. Now both the sugars, the glyceraldehyde 3-phosphate and DHAP, both are interconversible form, and with the help of triose phosphate isomerase enzyme, they both get interconverted into each other. Now we come towards the second phase of glycolysis. In first phase of glycolysis, the energy demanding phase or energy requiring phase, because the 2 ATP are required. But when we study about the second phase of glycolysis, you remember, it is energy generation phase, or the energy is produces. Energy in this phase, you remember, produces in the form of NADH, as well as at the 2 reaction or at the 2 sites ATP is also produces. So energy generation phase is the second phase of glycolysis, where the energy is produces in the form of NADH as well as in the form of ATP. Now here glyceraldehyde 3-phosphate, first 1 molecule of glyceraldehyde 3-phosphate is entered. After that DHAP again isomerized in G3P, and again that G3P is also undergoes in the same pathway. That's why every time we multiplied 2, because every time the 2 molecules are one by one the cycle pathways take place, but at a time 2 molecules of the same are formed. So here you remember glyceraldehyde 3-phosphate now catalyzed by the G3P dehydrogenase or glyceraldehyde 3-phosphate dehydrogenase. This is the reaction where energy is produced in the form of NADH, and 1 molecule of NADH, you remember NADH plus H plus लिखते हैं हम लोग. So 1 molecule of NADH and H plus, you remember, produces a 3 ATP. Now here you remember the G3P will change into 1,3-biphosphoglyceric acid. 1,3-biphosphoglyceric acid now reacted and by the phosphoglycerokinase, and due to activity of phosphoglycerokinase ये convert किस में हो रहा है 3-phosphoglyceric acid. तो यहाँ reducing form में याद रखिएगा decreasing order 1,3-biphospoglycerate, 3-phosphoglycerate and then 2-phosphoglycerate. So 3 reactions at a time you can recall. So 3-phosphoglyceric acid, again the 2 molecules are formed and it is reacted by phosphoglyceromutase. So the 3-phosphoglyceric acid become convert into 2-phosphoglyceric acid. Now when the 2-phosphoglyceric acid, one of the most important enzyme of the glycolysis and the enzyme which responsible for the release of water, this question is asked already. So enolase, you remember enolase is an enzyme which responsible for the release of one water molecule during the reaction, and secondly enolase is activating on 2-phosphoglycerate or 2-phosphoglyceric acid. Now due to the activity of enolase, the 2-phosphoglyceric acid get converted into phosphoenol pyruvate, or simply phosphoenol pyruvate is written as PEP. So 2-phosphoglyceric acid now become a phosphoenol pyruvate, and this phosphoenol pyruvate, again this is the important, so 3 enzyme you remember for the glycolysis. Aldolase for the breakdown of fructose 1,6 biphosphate into G3P and DHAP. Enolase for the formation of phosphoenol pyruvate, and the last enzyme of the glycolysis is pyruvic kinase. So enzyme which responsible for the formation of pyruvic acid, that enzyme you remember pyruvic kinase, and by the activity of pyruvic kinase, PEP or phosphoenol pyruvate get converted into the pyruvic acid. These are the reactions when the molecules are converted as an energy molecule. That is, in this reaction NAD become a NADH and H plus. Here the ADP become convert into ATP, and in this reaction, last reaction of glycolysis is also, you remember, energy generation phase or energy generation reaction, and it also responsible for the production of 2 ATP. So in energy generation or in second phase when you go or when you appear, then you remember the one molecule of NADH produces by glycerol DI3 phosphate, and the second molecule of NADH is produces for DHAP when DHAP convert into glycerol DI3 phosphate, and again it undergoes in the same pathway. Similarly, the 2 ATP are produced at the formation of 3-phosphoglyceric acid, and the 2 ATP further produces in formation of pyruvic acid. In this way, when we study the energy generation in glycolysis, so in energy generation of glycolysis, phase 2 produces 2 molecules of NADH and H plus. These 2 molecules of NADH and H plus responsible for the formation of 6 ATP, whereas at 2 sites, this site and this reaction and this reaction, responsible for the generation of 2 ATP. So total 4 ATP are produced. So in this way, in second phase of glycolysis, the total 10 ATP are generated, but when we study about the net gain means out of these 10 ATP, 2 ATP are utilized in phase 1, so that you remember the total how many ATP are formed. Total or net gain of ATP in terms of energy currency, you remember, for glycolysis is 8 ATP. So this thing you remember, the pyruvic acid, now you remember the pyruvic acid which is formed. This pyruvic acid is originally synthesized in cytosol, and in anaerobic respiration, this is the only pathway for the oxidation of the glucose. But in aerobic respiration, when the organisms are aerobic, and they show the aerobic pathway for respiration also, so this pyruvic acid now enter into the perimitochondrial space. Perimitochondrial space कहाँ पे available है आपका? The outer chamber of mitochondria is called as perimitochondrial space. So now in aerobic individuals or aerobic cell, this pyruvic acid enter into perimitochondrial space, and there it involve in the link reaction. So that we will study in our next session. So students, you revise this glycolysis part number of time, and repeatedly you go through the chart, because the questions mainly related with glycolysis are asked on the basis of this chart. So you prepared this chart very well and revise this chart. If you face any difficulty, तो mostly आप first time में phase 1 को revise करिए, second time में phase 2 को revise करिए for first 2-3 time. After that जब आपको chart prepare है, after that you revise entire chart at the same time. Okay? तो यहाँ पर आपको glycolysis से related in case अगर कहीं भी कुछ doubts है, तो वो doubts आप comment box में लिख सकते हैं. And I think as well as I hope that whatever doubts you are face in glycolysis, that maybe you recover by this session. Okay, and by that time you stay blessed, stay motivated and stay positive.